Hello or welcome to Ferdinand tutorials. In this video, we want to talk about reverse transcription. Now, we're not going to be talking about this reverse transcription. We're also going to be talking about the enzyme, which is a reverse transcriptase. Remember that in your genetic dogma or your molecular biology dogma, that's why we say that information moves from DNA to RNA and from RNA to protein. The first, which is transcription, and the second, which is what? Translation. That means when information moves from DNA to RNA, it is termed transcription. And when information moves from that RNA and translated to protein, it is called what? Translation. Now, today our focus will be on information that is moved from DNA to RNA and from RNA to DNA. I don't know if you are getting what I'm saying. So normally, when information moves from DNA to RNA, it is called transcription. When information moves back from RNA to DNA, it is called what? Reverse transcription. That is, and when we are talking about genetic dogma, we say that information cannot be moved when it have gone through this DNA to RNA to protein. It cannot move back. But the exception of this rule, there are two exceptions, where we have the prion protein and we have the reverse transcription. So I want to be talking about this reverse transcription. Now, reverse transcription is the process whereby RNA information are moved from RNA to DNA. Whereby reverse transcriptase, which is my main case story today, is an enzyme used to generate complementary DNA. That's the cDNA from an RNA template through a process called reverse transcription. Reverse transcriptases are used by viruses such as HIV and hepatitis B to replicate their genomes by retrotransposition, a type of genetic component that copy and paste themselves into different genomic locations by converting RNA back into DNA through the process reverse transcription, using an RNA transposition intermediate. Contrary to a widely held belief, the process does not violate the flows of genetic information as described by the classical central dogma, the dogma I explained earlier, as transfers of information from RNA to protein are explicitly held possible. Retroviral reverse transcriptase has three sequential biochemical activities. First activity is RNA-dependent DNA polymerase activity. Second is ribonuclease hedge, that's RNase hedge. And the third is what? DNA-dependent DNA polymerase activity. Collectively, these activities enable the enzyme to convert single-stranded RNA into double-stranded cDNA. In retroviruses and retrotransposons, this cDNA can then integrate into the host genome, from which new RNA copies can be made via host cell transcription. The same sequence of reaction is widely used in the laboratory to convert RNA to DNA for use in molecular cloning, RNA sequencing, polymerase chain reaction, that's PCR, or genome analysis. Now let's talk about the application of this RNA transcriptase, because we cannot say there is no application and we are studying this way. No, there must be an application. There must be something that it does. So let's talk about the application or where it can be applied to. Now, first is what? The antiviral drugs. As HIV uses transcriptase to copy its genetic material and generate new viruses, part of retrovirus proliferation cycle, specific drugs have been designed to disrupt the process and thereby suppress its growth. Collectively, these drugs are known as reverse transcriptase inhibitors. We've heard of inhibitors, something that inhibits a reaction, something that prevents a reaction, something that puts stop to a reaction. So it does what? They are known as what? Reverse transcriptase inhibitors, and include the nucleotide and nucleoside analogs. We have example, the zidovudine, we have lamivudine, etc. There are many of them. So the second application here is molecular biology. Reverse transcriptase is commonly used in research to apply the polymerase chain reaction. Polymerase chain reaction is a method widely used to rapidly make millions to billions of copies of a specific DNA. Now we say a reverse transcriptase is commonly used in research to apply the polymerase chain reaction to RNA in a technique called reverse transcription polymerase chain reaction. So that is all for this video. That is the little we can give you about reverse transcription and reverse transcriptases. So thank you for watching this video. I'll be seeing you again on this channel on Thursday try and be here, try and be present. We will be giving you a new package for another teaching, another video that will interest you. Thank you all.